everyone. Thanks for having me. So as Bill said, my name's Amy, and I'm here up on the 6th floor. And today I'm going to talk about some of the work that we've been doing on population mobility and how this can be related and impact malaria elimination. So this is the map of malaria endemic and places that are controlling malaria in the 1970s. And importantly, at this time, no countries were aiming for malaria elimination. Since that point, that map has changed quite a lot, and there's a number of countries that are now aiming to eliminate malaria at some point, at some time divided by 5. And the reason you need to think about importations is it becomes important when thinking about elimination, because elimination is really only going to be plausible in places where you're not at risk for having constant reintroduction events. So what those reintroduction events actually mean can vary depending on the setting, but you definitely need to start thinking about not just sort of are we in a low transmission setting, but sort of what's the risk that malaria may come back if we're able to control it, and if so, is that going to impact our elimination efforts? And there's a couple of reasons why this is important. So one is resurgence events. So this is adapted from Justin Cohen's paper that shows what year all these different resurgent events occurred. So these are primarily countries that are either eliminating or controlling malaria, where it was able to lead to secondary cases after international introduction events primarily. Another reason is thinking national scales. So you might be able to really control malaria in parts of your country, but if you have a lot of travel, you might actually have source populations within a country that reintroduce the disease. So this is a map that's from the 1960s by a report that shows different migrant labor routes, and what he was able to do is tie this with different source populations and sources of migrant labor that then led to epidemics across Tanzania. And finally, you need to think about introduction events and human mobility to try to think about targeting different interventions. So this is particularly the case in Asia, where you see a lot of clinical cases at health facilities or community health workers in villages, and so you might think that you need to target some vector control here, but what is actually happening is people are traveling and working or migrating into forested areas that are known to be transmission hotspots. And so although you're seeing your clinical cases in this location here, you should actually be targeting where people are getting infected. And broadly, when we think about malaria importations and how it might relate to malaria elimination, we try to think about both, where will there be introduction events? Are they frequent, infrequent? Where are they coming from? Are there ways that we should be coordinating efforts within a country, across borders, about where there might be source populations for these introduction events? And sort of operationally, how can we actually use this information to try to figure out either where should we be targeting interventions, or where should interventions be coordinated amongst locations? And so there's a lot of work that's been done about sort of this idea of what counts as local versus imported cases. So this is part of the E8 in Southern Africa for a number of studies where they sort of estimated how many of those cases are either imported locally from the same country or nationally. And so you can see it's quite variable. So if you go to a place like Lusaka in Zambia, in a hospital there, almost all of your cases would be coming from outside of Lusaka. In other places, you may only have sort of 5% of your cases are being imported. But being able to do that, and it also matters sort of how those importations are occurring. So do you think that you have a situation where you have these number of importations up top that are leading to some of those local cases, versus you have really one importation that's leading to multiple secondary cases. So trying to distinguish about how you separate those off and how you identify these different cases is relevant for elimination. But all this implies that you're able to measure these things. So it all sort of implies that we can measure travel or mobility, either by infected individuals, or we can measure the movement of the parasites. And so what we've been doing is trying to surround the problem of trying to look at a combination of human mobility data and parasite genomics to try to figure out how are parasites connected, how are different populations connected, and how can we measure these movement patterns. And so there's lots and lots of different ways that you can quantify human travel patterns on different spatial and temporal scales. There's a lot of population-based data sets like international flight data, migration data, or census data. You can also do individual level data like travel surveys or GPS loggers. And all these different kinds of data sets will sort of cover the temporal and spatial scales that you're trying to get at. But as with all data, there's lots of biases, there's always going to be lots of issues. So for example, GPS loggers might give you very detailed data, but it might only be about a few individuals. Things like a census or international migration data might not be frequently collected, and other types of data sets may not be available in countries you're interested in. For example, here's census migration data. So this is a map that shows when the most recent census was collected that has an origin and a destination location. So in Pakistan, for example, it's the 1970s. So one could argue it might be a little bit outdated. Even when the data is available, it's often at a very coarse temporal scale. So this is the most recent data that's available for migration between these different, in sub-Saharan Africa. And for example, you'd say that there's no importations ever into Madagascar, or not. So even when the data is available, it might not be very granular. And when thinking about malaria control in countries where it's very heterogeneous, it sort of matters if the person's coming where there is malaria transmission ongoing, or if they're all just going to the capital city where there might not be any malaria. And other types of data, like flight data, might have a very poor resolution where malaria is endemic. So this is just a plot that has population size versus the number of international airports. So most of the airline data relies on international flight patterns. But for example, so in the red line is like the smooth line for Africa, there's not a huge number of international airports. This could also be why no one ever predicts that influenza comes to Africa, where there's just not enough airports to be able to have really granular data. So what we've been using mostly is trying to use mobile phone data and work with telecom operators to try to understand human mobility patterns on national scales for millions of individuals in these countries. And the way we do this is we use these data, which are called the call detail records, that record who's calling, who they're calling, and when it's occurring. This is data, an example data set from Norway, and it's my friend Kent. He said we could have the data. And the way that we analyze these data are, if you have the locations of the different mobile phone towers, what we're following is different anonymized subscribers who are making calls or sending texts or receiving them at different mobile phone towers. Then what we do is we aggregate them up and say which mobile phone tower they use the most in a given day and where is that. And so this person, this subscriber, we're going to say was at tower C on day 1, and then they're moving around. And the way we measure mobility are when between subsequent days when people are changing their location or their most used tower. So this allows us to get an estimate about the movement patterns within countries where there's mobile phone coverage for millions of individuals. So there's lots of biases with these data. If there's no mobile phone towers, we can't do anything in those situations. So particularly in very, very remote or rural areas, there may not be any coverage. And it's also biased by ownership. Not everyone owns a mobile phone, and so we're definitely missing certain populations, particularly children. Nonetheless, we collect these data and are trying to use them for a bunch of different pathogens across sub-Saharan Africa and Asia. So what we do is we take these data and we identify some sort of measure of mobility or connectivity between populations, and then we combine this with a really simple model to try to identify where are people moving and where are parasites moving. So sources are where parasites or people are coming from, and sinks are sort of where they're going. So this is work from Madagascar, where we can look at sort of where the sources and sinks of travel are, and where the sources and sinks of parasites are, and importantly, try to identify where are different places that maybe have the same sourciness, but are sending their parasites to lots of different places. So in terms of targeting interventions, you're going to have a different impact if you target different types of locations based off of the travel patterns and where those people are traveling from. But these data might not actually be capturing who's infected. So we definitely play like the law of large numbers, that we have like millions of subscribers in these data sets, but it's sort of a very coarse way of looking at it. Sort of on the opposite end of the spectrum is trying to look at travel surveys, where you may have symptomatic individuals who report travel, and if their travel is to a malaria endemic place, then you'd say it's imported, otherwise you may say it's local or not. But people aren't perfect. They can't recall everything. And importantly, you may be missing asymptomatic infections, who, based on whom may or may not matter or not. So what we've been trying to do now is trying to answer these broad general questions using a combination of data sets. So each one of these data sets have their own particular biases, but maybe if we combine them all, it can kind of combat some of those. Travel surveys are very specific. The person had malaria, where they recall where they went. You can ask them very specific questions, but you're kind of going to be missing a lot of people too. Mobile phone data is sort of on the other end. It's anonymized, it's millions of people, so maybe it's sort of bounding the problem in some way between those. So I'm going to talk a little bit about two studies, one in Namibia and Bangladesh, with Brian and Sophonius in Namibia, and with Caroline Bucky, Shahan Chang, and Richard Maud, mostly based out of Harvard and Bangladesh, where we did two very similar types of studies of collating all these different data sets. So we have mobile phone data, we have a geographic model to look at importations, we collected travel survey data, and we also did parasite genotyping. And both of these countries are in somewhat of a similar situation where they have lower malaria incidence, but they border higher malaria incidence places, or within the study areas, there's also high malaria incidence. So in both situations, we're really trying to understand what's the utility of these different data sets, can they be combined, and how can they sort of identify where people may be getting infections. And so both studies worked where symptomatic individuals were at a health facility. We have mobile phone data that covers most of those health facilities. RDTs or DBSs were taken from genetic data and then travel history. So briefly, some of the data sets. So these are the mobility data sets where we have mobile phone data from Bangladesh and Namibia, and we also did travel surveys in each of those countries. So these are just some results from the Bangladesh study. So in Bangladesh, we really just focused on this Chittagong Hill Tracts region, which is where most of the malaria is. And we can map sort of where are people traveling based off the travel surveys or based off the mobile phone data. So one thing to note is that these data sets spatially don't necessarily overlap a whole lot. There's not a huge amount of coverage in the forested area where we specifically asked people if they had traveled. But we can also look about sort of what are we saying about the proportion that are imported or locally acquired based off these different data sets. And if you squint really hard from quite far away, there seems to be more importations on that bottom left-hand corner. So we did a similar study in Namibia where we also have the mobile phone data and the travel survey data. And here we just focused on these two different areas in Kavango and Zambezi in that corner strip of Namibia. And once more, sort of like the mobile phone data is estimating a lot more travel. It's estimating a lot more travel between Kavango and Zambezi than the travel survey data that would suggest there's a lot more local travel happening. So, we can measure the human using travel surveys or mobile phone data. And there are sort of pros and cons of using each of these different types of data sets. Travel survey data, people may have recall biases, but you can ask very specific questions. So if you want to know if somebody went to this particular place, you can just ask them in the travel survey and get information on that. Mobile phone data, you get big numbers, you can look across entire nations, but not everyone owns a phone, and importantly, not everyone who's making calls may be the ones that are transmitting parasites. So what we also wanted to see is, can we just measure the parasite directly too, using genotyping? Genotyping, it may be informative in these particular types of low or medium-low transmission settings. It's becoming cost-effective, but we sort of needed to develop new tools that you can try to combine these different data sets. And I think there's still a big gap about how these types of molecular tools and other sources of data are actually translated and used. So in both of the studies, we have genetic data too, so they were sequenced slightly differently. So we used the 101 SNP barcode with Sanger in Bangladesh, and in Namibia, we used 26 microsatellite markers. And so we can identify highly related parasites from the genetic data. So these are the plots that show the distance versus the proportion of highly related infection pairs from genetics in Namibia, so the Kavango East region and the Zambezi region. And from the genetic data, it would suggest that most of the transmission is occurring locally. But there does seem to be some sort of quite far out in terms of distance, highly related parasites. And we can combine this. So the mobile phone data and the travel survey data, we can get a sense of sort of where do we think importations are occurring, which places do we think are connected or not. And we can develop a measure about like the percentage of importations in each of those locations. And what we can do is we combine that with the genetic data to say there's also genetic evidence, either that we have highly related parasite populations that are far away, and try to figure out sort of where is that occurring frequently or not, and are those the same places or not. So what we roughly find is that the mobile phone data and the travel survey data, compared to the genetic data, seems to be identifying similar locations. So where there's evidence of mobile phones that, lots of parasites are probably being imported there, we also see evidence that those places seem to have highly related parasites that are further away. But there is also a lot of noise between those two different types of data sets. And we do see fairly similar results between Bangladesh and Namibia, that on big picture, broad scales, they can identify some similar locations. But ultimately, we want to try to say something on finer scales and be able to identify sort of how related these data sets are. And importantly, can things like genetic data sort of help supplement some of the issues that are in the human data that we're measuring travel for. And so we do find that with the genetic data, you get quite, you can see a much finer resolution than the travel data, and you can say things about larger geographic distances. And so in the Namibia study, we've also looked at cross-border travel that Brian Greenhouse will talk about in the afternoon. And in Bangladesh, we've been trying to sort of combine these data sets too and try to see sort of where's all the different data sets overlapping. And can we sort of make some sort of composite measures of where we think that these are places with high parasite connectivity? So pretty much what we did is we just combined the different routes of travel that resulted in a large number of importations estimated from either the mobile phone data or the travel survey data. And then we just overlay that with the locations where we think there's a lot of genetic evidence that parasites aren't from there. And what we're trying to do is figure out sort of how is this comparing to what the National Control Program would be thinking in terms of where they need to target. So on that right-hand figure, the high incidence areas are the ones that it's thought that most people are getting malaria, and it's where treatment and intervention should be targeted. But if you try to look at sort of where parasites might also be moving around or imported, that's giving you a slightly different picture, where you also need to be focusing on a lot of these high population places, which maybe aren't causing a large amount of, which can cause a large amount of cases because you have a large number of people traveling. So we're trying to summarize and figure out sort of what data set should be used, what's the benefits of each of these kinds of data sets. So if we try to break apart the question is, are you able to identify a local case as local? And are you able to identify an imported case as non-local? The different data sets are going to perform well in different instances. The parasite genetics, if it's local, it will have a very strong signal, but it may be missing a lot of cases. The mobile phone data is really going to be overestimating cases. It's really going to be saying a lot of cases are probably imported, and a lot of those people might not even have malaria. And travel survey data maybe will go somewhere in between the two. And so there's ways to improve. If ultimately we want to try to get up into that top corner and have the best combination or type of data set, there's ways we can improve each one. So travel survey data, we can, if we can reduce recall bias or be able to survey both symptomatics and asymptomatics, we're going to be able to improve the utility of that different data set. The parasite genetics data, if we have representative parasite genomes, we might be able to, on a more granular scale, be able to say, this one's really not from here, this one really is from here. And the mobile phone data, if we have more ideas about bias and who we're actually measuring and better models of trying to look at when people might be getting infections, we might be able to increase that data and improve that data by itself. But the parasite genomics is also able to improve the types of data, information you're getting from the types of survey data, and vice versa. So it actually might not be true that one data set is going to be the one that does it all, but maybe a combination of the different data sets and trying to identify what's the biases in each one, and is it a bias that can be helped in some way by another type of data set that also has a lot of biases. But I think it's also important to note that, it's really going to depend on the transmission and scale that you're looking at. So if you're in a really high transmission setting, the parasite genetics might not give a refined enough signature, so you might be in a completely different space than this. This is very much guided by our work that's in these low or low to medium transmission settings. But if you're trying to do this in high transmission settings, I really don't know what you should do. But definitely get a lot of data. That's not a bad idea, but I don't know how to analyze it. All right. So just to summarize a little bit, so importations are an important thing to think about in terms of malaria control and elimination, and there's lots of different ways you can do this, including newer types of data sets. But all these data sets are going to have different biases and may be useful in different situations or not. So we sort of think that some way about combining all these different data sets, and trying to not say, this data set says this one thing, but where do you have multiple lines of evidence all sort of agreeing in particular locations or not? It may be a more robust and useful way to try to identify where importations are occurring, where they're coming from, and how intervention should be targeted. So there's many, many different people who are involved in all of this different work, and a number of different funders, and that's an old slide. Okay, thanks very much.
Thank you, Amy. That was fantastic. The questions? Yes.
Hi, Amy. Thanks for that talk. That was really interesting. And I'm actually sort of making a plea for a very pragmatic use of these data as well. So we know that a number of different interventions that we have in elimination settings or to accelerate towards elimination probably don't work as well as they could because of high population mobility. And it's not just an importation issue, it may be a coverage issue because people are gone, they're not there to take MDA, et cetera. And we make recommendations that say things like limited risk of importation, and yet we have no way to quantify that. Nobody in most of these studies is able to measure, we don't have any common index for mobility from which we can begin to compare studies and get a sense of how much that mobility may have compromised the intervention. So I guess to the general community, I would just make a plea, if there's some way that you could also use these data to start helping us come up with good indexes that could either be measured through mobile phone data or in some other on-the-ground way that's comparable between studies and allow us to begin separating out this impact. Thanks.
We can. Maybe Saturday. Tomorrow I have to teach class.
Hi, Amy. Thanks for the very nice presentation. I'm Shannon Takala Harrison. I'm at University of Maryland. My question is about kind of the temporal scales for the genetic data. Like, I guess, can you measure how temporally relevant different markers are? So, for example, the barcodes, those are common SNPs. They may reflect an older signal, or if you're looking at haplotypes, you may be able to capture a more recent signal. Or can you comment just a bit on the temporal resolution of the genetic data?
Yeah, I think Brian can definitely talk about it more than I can, but I can talk a little bit. Yeah, I think it really depends what kind of genetic data you're getting, and it's also not clear to me, so if you try to do things like haplotypes, which maybe you're going to have high diversity and really be able to tease apart the transmission maybe, but then as soon as you're in a higher transmission setting, is that almost too noisy or not? So I don't really have a good answer about sort of which types of markers are the most useful in different things, but I think trying to also think about what transmission setting are you looking at and what time scale of mobility you're trying to measure, and if we can also try to use some simulations to try to get at how informative do we think different markers would be or not. I don't think anyone's really solved it yet, but I think it's a really important part because sometimes you do it and you see no population differentiation, and how much of that is how you're sequencing and what you're trying to look at, and is it mismatched with the thing you're trying to measure or not. But how is that also different if you went into a different transmission setting or not? So I don't know, Brian, do you have any other things to add about it?
That was perfect.
Thanks, Brian. Yeah, but I think it's an important thing because also if we're trying to make a push that genetic data can be useful in these settings, well, we need to know in what settings and what type of genetic data.
Any other questions for Amy? All right, thank you very much,
Thanks.
Amy.